Good evening and welcome everyone. My name is Ana Rubio and I work in the Natural Resources branch of Hornsby Shire Council. We are here today collaborating with Hornsby Library on this event called Spotlight on Science. I would like to start by acknowledging we are meeting on the traditional lands of the Diagn and Hornsby people, and I recognize the continuing connection to land, culture, community, and story. I pay my respect to elders, past, present, and emerging. So thank you everyone for joining tonight this event and to celebrate National Science Week. We had a really well attended and successful event as part of last year's Science Week, and we are hoping to repeat that today. The goal of this event is to learn from some of our local scientists that they live in the Hornsby Shire, what inspired them to get into science, and maybe some tips for our younger listeners who might want to become scientists too. So we had quite a diverse set of scientists tonight in the panel. I am too a scientist. I work with a number of environmental scientists in Hornsby Council, and specifically I work looking after the Hawkesbury estuary. I started my science career in Spain, that's where I'm from, and I then moved to the UK where I further studied fisheries and aquaculture, did my master's work there, and then came to Australia where I did my PhD working with the oyster industry in New South Wales. So through my career, I've been in beautiful places, I've been working with amazing people. So I look forward to listen from our panelists to see what stories they can share with us about their career in science. So each panelist is going to give a brief talk, and then we are going to have the Q&A session. The way how you can contribute from home is by clicking on the Q&A icon that should be somewhere on your desktop, and a window will pop up, and you will be able to type your questions there. At the end of the 4 talks, we will open the Q&A session and we will try to answer all your questions. So let's welcome our first panelist. She is Bethany Ellis. She is a paleoclimatologist working in the Australian National University. And last time that I spoke to her, she was finishing her PhD thesis. So I'm looking forward to know if she's actually finished, what it drove her to do even a PhD, and to hear anything that Bethany wants to share with us. Welcome, Bethany.
Hi, thank you very much, Anna. I'm just going to share my screen. Hopefully you can see my screen now. Yep. So hi, I'm Bethany. I'm, I guess, a PhD student still at the moment. I've submitted my thesis earlier in the year and it has been marked, and I'm just working on the thesis corrections, which is the little tweaks to finish it off before I can officially graduate and hopefully graduate in person. So I'm doing my PhD down at ANU, but I did grow up and have lived in Hornsby Shire pretty much my whole life. So yeah, I'm really pleased to be here today. So I'm going to start with my journey from high school all the way through to where I am now. I've always loved science. In high school, I did all the sciences that I could. So for my HSC, I did do chemistry, biology, earth and environmental science, and then you're not allowed to do a 4th science, so I had to choose agriculture instead. But I absolutely loved all the sciences. Earth and environmental science was my absolute favorite, and I'm so glad that I had the opportunity to do that, because I definitely think that is what led me into the career path I am in today. During undergrad, so I went to the University of New South Wales in Sydney. I did all the earth and marine science courses that I could do, particularly anything that involved fieldwork, because I loved being outside and doing science, you know, putting it into action in the field. I still did enjoy lab work, but there's nothing beats being like out on the beach doing work rather than being stuck in a classroom all day. This led me to travel all over Australia. I did work all across New South Wales looking at the geology of the Sydney Basin, so everything from Newcastle all the way down to the fossils of the Ulladulla rock platforms. I was fortunate enough to go up to the Great Barrier Reef, and I did 2 weeks of field work up on Heron Island, swimming with the fish and the coral. That was definitely a highlight. I was also fortunate enough to do a research cruise from Hobart across the bottom of Australia to Fremantle. That was 2 weeks where we were mapping the seafloor and analysing ocean chemistry. It does get a bit rough and I did get seasick, but I still absolutely enjoyed that too. So I finished my undergrad with a Bachelor of Science majoring in both marine biology and marine geology. And I also did a lot of spatial information systems, which is sort of things like satellite imagery and GIS. I then did my honours also at UNSW, and that was in climate science. And I'll touch on that a little bit more too. As I said, I love to travel and be outside. So this is some of the photos from my fieldwork, snorkeling on the Great Barrier Reef, down at Bondi Beach measuring the beach before and after a storm is really important, understanding coastal erosion. I did trips up to the Smiths Lake where we looked at the fisheries within the lake. Southern Ocean, there's not a lot to see when you're on the boat in the middle of the ocean, but there is a lot of scientific data to be collected while you're out there. And so when I finished my undergrad, I had a love of both earth science and marine science, and it took me a while, but I managed to find an area of science that kind of combined both of those, but still for me had a purpose, and that was paleoclimatology. So paleoclimatology is the study of past climates because instrumental records really only go back 50 years at most, sometimes stretch back 100 years for some parts of the world, but we really don't know a lot about the climate before about the 1960s. So paleoclimate is really important because we use natural archives to reconstruct the past climate. This helps us understand the natural variability of the climate before humans started having an impact and how the anthropogenic impacts compare to the natural variability. And when I talk about natural archives, I'm talking about things like ice cores and coral skeletons, tree rings, the sediments at the bottom of a lake or the bottom of the ocean, and also speleothems, so deposits that we find in caves. And you'll notice these photos, which are all slices of these natural archives, are all really layered. And the layers are really important because things like coral grow every single year. They add a new layer to their skeleton. And as they grow, they incorporate environmental information into their skeletons. And I thought this is absolutely fascinating to know that you could reconstruct, monthly temperature data from a coral skeleton going back hundreds of years to look at what the climate was doing in, say, 1883. And so this is kind of the area that I fell into. It involved a good amount of lab work, computational work. If you're lucky enough to do field work, you can get out into the field. Unfortunately, I did miss out on the field work, but I did get to travel to some amazing places anyway. So my honours research was in ice core paleoclimatology, and this was done at UNSW. Unfortunately, I didn't get to go to Antarctica because the samples were collected before I go. A lot of our work, you have to collect the samples usually in advance of the project because by the time you get them through quarantine, your year is almost done for honours. It's only 8-month project. You don't have months to wait around for samples. So we use biomarkers, which are things like the proteins and amino acids in cells of biological origin. And we looked at these in the Antarctic ice. So these two people here are walking across the ice. You can kind of make out little holes in the ice, and this is where they've taken samples. These samples stretch back over 15,000 years, and each sample they took, we then looked at biomarkers in the ice. And this gives us an idea of how much sea ice extent there is around Antarctica. So the sea ice is the ice that floats on the water around Antarctica. And the more sea ice there is, the less biomarkers, the less plankton that is being blown in from the ocean onto the ice because there's more distance between the Antarctic ice and the edge of the ocean, like in the picture on the right. And so, we discovered that, plankton is actually quite common. We found in these ice cores, and this was the first study of its kind. We also did ancient DNA to discover that there was a huge biodiversity difference between plankton back then and plankton now. And this does fluctuate over time. So it was a really exciting study that was published about a month ago. Even though I did the work almost 5 years ago, it was a big study with a lot of different scientists in. I then decided to jump into the tropics for my PhD research and ended up doing coral paleoclimatology. So using fossilized coral skeletons from Indonesia to reconstruct the climate of the Indian Ocean. Again, this was a lot of lab work, and I did miss out on going and doing fieldwork, but I did go to Indonesia for a conference. This beach that you see in the photo with the big rocks in front of it, all of those rocks are actually fossilized coral. Indonesia is quite unique in that during tsunami events, due to the earthquakes and volcanic eruptions, a lot of the coral reefs get quite badly damaged and the coral gets thrown up onto the beach by the tsunami waves. This allows us to go and travel back in time through these coral boulders to reconstruct the climate over several thousands of years. So even though each coral may only live for 100 years, a coral from 1,000 years ago gives us 100 years of data. This is really fascinating because it really does link into what we're seeing today in today's climate. The coral are easily drilled. We don't harm them if they're living. We take a core through the middle of them and then it's plugged, the hole is plugged back up and the coral keeps growing. The fossil coral just makes it a lot easier to access older material because it's thrown on the beach and it just sits there until we come along and drill into it. Again, these images of the coral can show you the layers, and each layer represents, they grow a new part of their skeleton every month, so we can get monthly records back over hundreds of years. Just like humans, you can record, record where people have been living based on the isotopes in their teeth. The same thing applies for coral. And so the main question of my thesis was, how has the Indian Ocean Dipole behaved over the past 1,000 years? I think many of us who have lived in Hornsby Shire, especially over the last 20 or so years, will have seen and smelt definitely the bushfires that we've experienced. So back in 2000, we had some really strong bushfires in Hornsby. And it's bushfires like the ones we've also just experienced last summer that are largely not caused by, but the drier conditions we experienced were resulting from the Indian Ocean Dipole, which is the climate system of the Indian Ocean. And so, you've got reduced rainfall over Australia, which leads to drought and bushfires. And so I think as a climate scientist, it was hard to study this whilst we're watching, the country burn. But it's also, I think, a very important area of science to study. And just one last thing I'd like to share is, I think earth science is often very overlooked when people go to decide on what to study at university or what to study in high school. Earth sciences is not just geology, it's so much more than that. It's not just mining and energy resources. Earth sciences often gets a very bad public perception, but there is a lot going on in earth sciences. You get amazing opportunities to travel all over the world. I believe that earth scientists are the key workers for the climate crisis, just like medical workers are the key workers for the COVID crisis. There's a lot of different job opportunities. We are looking at global solutions to have a sustainable planet. And coming up into the future, there's a lot of different areas of earth sciences which are being expanded into. So the Green Stone Age, for example, is utilising more elements from our periodic table to be able to sustain green technologies instead of using our traditional coal and oil resources. You have the digital earth, so we're collecting so much data about our earth, and we need to look at, all the data and how we can monitor it and look at natural hazards and how the earth is changing. As cities expand, more infrastructure is being built underground, like the North Connects tunnel that's just down the road from us. That wouldn't be built without earth scientists being able to work out where the best place to drill is based on the underlying geology and the faults. And also food security. So we do a lot of, earth scientists do a lot of work on water storage and the geochemical interactions of the Earth's surface to make sure that we can, produce enough food for us and in the future as well. Thank you very much.
Thanks, Bethany. That's fascinating, amazing photos. I look forward to see what questions come up. So our second panelist is Associate Professor Nathan Langford. He's a physicist and a quantum engineer, and he works at University of Technology, Sydney. Thanks, Nathan.
Well, hello. Thank you very much to the organisers for inviting me to participate. This is great, a lot of fun. And so I just wanted to tell you a bit about my perspective, where I've come from. I thought I'd do it kind of in reverse order this time. I'll start telling you a bit about what I do and for some context, and then tell you a little bit about how I got here at the end. Really, I guess that will mostly come up in questions and so forth. So basically, as Ana said, I'm a physicist, an experimental physicist, and also something that's called a quantum engineer. This is really a career that has only existed in the last couple of years. I mean, prior to that, nobody had ever heard of this as a field. So let me tell you a bit about it. Oops, I'm not getting, I need to click. Okay, so what is quantum computing? Quantum computing is something that you may have heard about. There's been a lot of news stories about it. If you ever read tech pages, then it's all over the tech pages. It's really getting more and more interesting reports, particularly something that, if I start off my fancy laser pointer here, you know, last year there was a lot of interest as Google got to something called quantum supremacy, but really there's work being done by like huge numbers of tech companies from Google, IBM, Microsoft, Intel, Alibaba, Huawei, Tencent, Honeywell, and Amazon is doing it. This is something that's really now, you know, when I started, it was a research field only, and it's something that's really now kicking off completely into the industry area. Okay, so there's a lot of talk about it. There's now a lot of government funding about it. We just, you know, from China, billions of dollars being spent from China and the US and you know, in Europe and everything. Recently, we started up in Sydney, something called the Sydney Quantum Academy, which was a big investment from the government to try and capitalize on the fact that actually, surprisingly, in this field, Australia and particularly Sydney are really global leaders in this area of research. And we have been doing absolutely world-leading research for the last 2 decades. And so there's a big investment in the Sydney Quantum Academy to try and help build on that to make something really special. Okay, so there's a lot of people talking about it, but what is quantum computing anyway? Well, to start understand quantum computing a little bit, I won't tell you much about it, but you have to go back to a normal computer, what we call a normal computer. Sometimes we call classical computer, which is different from quantum. And here we have a picture of a kind of a silicon chip. And the point is that a computer is built of ones and zeros, these things called bits. And everything at the heart of a computer is in these, encoded in these ones and zeros. And basically everything that a computer does is designed around how to encode information in these ones and zeros, and then how to manipulate that information and by applying gates to these numbers so that you can control and process that information in a fancy way. Well, basically a quantum computer then, here we have a kind of a, you know, it's quantum, so we need to put it inside a massive ultra cryogenic fridge that is about 1,000 times colder than space. And here's a picture of the quantum version of this chip. This is a picture of the Google device that they achieved this amazing quantum supremacy result last year. And the difference between a quantum computer and a classical computer is that the quantum computer, the information is not stored in these bits, which are kind of very understandable, tangible, 0 or 1. It's like a light switch. It's either on or off. In a quantum computer, we encode this information in a quantum bit. And one way of thinking about that is you might have a quantum system that could be either pointing upwards or pointing downwards. But the thing about quantum mechanics is that these systems can occupy sort of much more fancy, can be in much more fancy states as well. So without going into any details about this, basically all the magic of quantum computing happens because you try and encode your information in a different way, and then you have to basically learn new ways of interacting with your information. So you have to learn new ways to control those quantum bits and play with them. So why would you want to do this is the next big question. My particular interest in quantum computing stems from something called quantum simulations. And this is an idea that was proposed now almost 30 years ago by a very famous physicist called Richard Feynman. And basically the idea is that if you want to, it turns out like quantum computing can do everything that a normal computer can do, but it's much harder to do it on a quantum computer. So you would only ever want to use a quantum computer if you're trying to solve a problem that normal computers find difficult. And it turns out that there are some particular problems that normal computers find extremely difficult to solve, where quantum computers can do it really well. Okay? And one of those problems is to, in fact, simulate the how quantum system evolves. So quantum mechanics is at the heart of a lot of our technology. And so understanding complex quantum systems is actually something that really underpins a lot of things in our society, a lot of technology. And so if we can, and it turns out that doing that on a normal computer is really, really hard. And there is a huge amount of supercomputing time, like a massive proportion of the supercomputing time worldwide is spent trying to simulate quantum systems on those supercomputers. And so if we could do that better, more efficiently, more fast with a quantum computer, then we could solve some really interesting problems, or at least study some really interesting problems. Like, for example, trying to understand how room temperature superconductors might work, complex materials. We could study in a computer drugs and pharmaceuticals and how they would work. So rather than having to always go and test things in the lab and build things into the lab, making some initial guesses maybe with a computer, but then we have to do things in the lab, we could start to really study those things on a quantum computer. And another thing is there are some chemical reactions that are really important. For example, there's something called the Haber process that basically is the way we create fertilizer. But it turns out that that uses a lot of energy. And so something like 4% of the world's global carbon footprint is involved with making fertilizers to feed people. Okay, and if you could do that better, you could make a big impact. So this is what we want to do. And what I do every day in the lab is some combination of plumbing. You can see lots of pipes here and connecting things up and pumping them around. Electronics with microwave electronics and soldering stuff. Fridge maintenance, which is this is what goes on inside this fridge. Basically, we have to put our experiments inside and design them really carefully. AutoCAD design to make the parts that we put in them. Nanofabrication, where we go into a clean room and make chips that look like this. This chip is around about, this thing here is around about 0.5 millimeter in length. And also computer coding and much, much more. So that's what we spend our days doing as an experimental physicist. UTS, I have I'm building a state-of-the-art quantum computing lab. This was what it looked like at December 2019, so we're really excited. This is what was supposed to be installed in March 2019 when we got our fridges. Unfortunately, then COVID happened, and so that's what's currently there. But this is nevertheless, that's what's waiting for us when we can get back in the lab and get everything installed. We're still waiting for the engineers to be able to come out from Finland to help us install these things. So just very briefly, what's my timeline? I started as an undergraduate student, and in fact before, at the University of Queensland in Brisbane. I did my undergrad, my honours, and my PhD all at the University of Queensland. And there in my honours and PhD, I was working with lasers and optical systems. So still doing quantum computing, but using lasers, things that look like this. And as part of my PhD, I was very lucky and got to go and do research at a great place in the US. And then I went overseas as a postdoctoral researcher, and I worked at some amazing universities in Europe. I spent about 10 years there. And then after all of that, I finally managed to get to the next point of getting getting a position, coming back home to Australia, and getting a position at UTS here. And now I work on these systems that we're talking about. So that's really just a very brief overview. I mean, here's the timeline, roughly speaking. there's a lot of stuff that you need, and we can talk more about this in the questions, but one thing that you do need, you need a lot of luck, and it takes a lot of time and persistence to do this sort of work. So you also need to make sure you're doing something that you love, and I'm sure we'll talk more about that in the questions later.
Thank you, Nathan. Amazing gadgets you play with there, and yeah, amazing the possibilities that are there with quantum computing. And completely,
Indeed.
that I can see what the potential could be. Just for those ones, before I introduce the next panelists, for those ones that they join us late, please feel free to enter your questions in the Q&A icon that should be somewhere on your desktop. Once we finish all the presentations, we will go through all your questions and we will get answers from the panelists. So our next speaker is Paul Gatsby. Paul has been working on conservation biology. He's a botanist. And I know that he's at the moment developing something extremely useful for me, at least, because I'm useless with identifying trees. So I hope that Paul is going to let us know about this little project. But Paul, welcome. And if you share your screen and take the microphone off, we can then hear your presentation.
Okay, welcome everybody. I'm going to talk about my project and a bit how I got there. This is a picture of me in the bush. When I think back and reflect back on my life, I really can see that the wilderness and the forest and nature really played such an important role in my life from very early on. I think my first memory as a kid is even just playing down by a river on our farm. And then, surprise, surprise, later after I'd finished my studies, hanging in the bush and meeting kookaburras, you know. So I feel so lucky to have that as my job, as my career. I think it really started. When I was back when I was a kid and I lived on a farm, we used to have like one channel on TV. It was kind of like pre-Google age for those who remember it. So it was a little bit different. I was always outside. And then I finished my studies. I ended up doing environmental science and a master's of conservation biology. And I just sort of fell into bush regeneration after working in a bar. So and I felt so lucky to be there in the bush every day, fixing up the bush. So I'll show you some slides of where I've been traveling and a bit of my work. So this is the Kapiti Valley. This is where I woke up one day. I was studying the trees there. There's some interesting species that only grow in the dry climates there. So my project is, I'm developing a book and an app. So it's a guidebook and it's focusing on the local eucalypt species. And so my intention with it is to really empower the community and get that connection between the land and the people in order to save these trees. Because I don't know if you're like me, you've zoomed out on Google Maps and you keep zooming, and then eventually the forest disappears, and it's a lot of our land has been turned to farmland. And certainly the New South Wales and Queensland are sort of world leaders, I guess, in land clearing at the moment. So it's something that really touched me after studying and growing up in the land, just to see massive changes to the landscape. So I thought I'd be quite proactive about it and start a project which I actually loved, and that was being out in nature, and that was combining education, and also it was adding this element of study, because in the natural world there's just infinite detail and infinite systems to really sink your teeth into. And for me as a career, I couldn't have stayed working at that bar. I kind of liked it. It was nice and social, but for me I really needed something that would stimulate me every day. And working in the forest, I've just found there's just, the more I learn, the more I don't know. So I feel really lucky there. So when I started this project, I was working full time, I was studying as well, and I realized how long it was going to take me to do this work and on this project. And work full time. So I did what everyone dreams of doing, brought a van, quit my job, and then drove to Queensland sort of thing, you know, to look at trees and ferns, and ended up in rainforests and doing all things like that, which was just an interesting life. And then eventually got back into Sydney once I realized how long my projects, I estimated one of my fern projects for a fern book was going to take 5 years. So I ended up back in Sydney working, and then really thoroughly getting my focus single-pointedly, not on a couple projects, but just on the one, the eucalypts. Okay. I just thought I'd throw in a little picture of my pooch there. This is Smoko. She comes on me on all the adventures. While I'm out under gum trees looking for gum nuts, she's out chasing bones. She thinks the gum nuts are a bit boring, I think. So within the Sydney region, there's a little over 100 eucalypts, and that's about almost 10–15% of what there is in the country. So it's very diverse, and I have been thoroughly interested in botany. I've always struggled, you know, with the eucalypts because some of them are so similar, and it's not really until you get a really good resource that it helps you understand what you're struggling with in terms of identification or knowing all the details about them. So I set out to create this book, and it's my goal is to have a book that's full of botany yet people can't put down. That's mainly plant people like myself, because there's a few of them that I have on my bookshelf which I've just read over and over again, you know, to absorb myself into them. So the book will aim to have about maybe 1,000 color photographs and descriptions and measurements of the trees in terms of where they grow, what makes an area transition from one type of tree, one ecosystem to another, and all various elements to the trees in terms of their height and their leaves and their gum nuts, etc. Okay, these are just a few of the pics what I'm putting into the book and the app. So I'm sort of shooting with a velvety black background just to make them all sort of float in space. That way I can move them around and put them close together. You can see in the middle, one of the flowers has been held up by a little clamp. So that's how I sort of work the magic, I guess. That'll be later edited out. And really, when you're looking at botany, there's so many important, minute details. So while I'm looking at eucalypts on the large, broader scale across the landscape, where they like to grow, the different variation within them. Also, I've got to zoom in and really sometimes using my microscope to look at these tiny little features on the gum trees and their flowers and the gum nuts, and that really suits me well with the sciencey part, like zooming in, having a look, and then zooming out just to get a really good overview. I get engrossed in this stuff for hours. So the picture on the left, that's a Angophora flower that's closely related to eucalyptus, and then there's mugger ironbark, and then there's a couple of new shots I'm working with of the leaves. They're really close-up macro shots where I'm shooting flashes behind the leaf to transmit light, and then that's why the leaf venation there, the veins running through the leaves, and the little dots are the oil dots that make the eucalyptus smell so good and also catch a light so well. And they're actually also really valuable identification features. This again is a close-up macro of the leaves, and I think even people without a eucalypt background can start to see some of the differences there, how tightly spaced those veins are, how many little dots are shining through. And you can see the two center leaves there. They're actually two closely related eucalypts. So their vein pattern is quite similar. So once you get into the little interesting nitty-gritties of the eucalypts, you can tell certain species or groups of eucalypts apart by their leaves alone. Okay, here's a little montage of mugger ironbark. This one is quite a pretty one because it has pink flowers or also red flowers. And the key here that I was aiming for with these shots is to really zoom in and show this feature because the top left has the buds, and the buds across all the eucalypts are slightly different. And the flower you can see in the top right, you can see on some of the little anthers there, they've got little dots and some don't, and that's a key characteristic, and that's really takes a trained eye to look at because that's quite a small flower, but it's a really useful identification aid. This is a whole selection of gum nuts. When I tell my friends I was working on a eucalypt book, they were like, "Oh, I thought there was only one gum tree," and I'm like, "No." You can see here there's a whole range. This is like 20 odd, and the size and variation there are just completely different, but it's really, once you have a close look at them, you can start to see some of the features. So this is about 20 of the 900 or so in the species and in the genus Eucalyptus. So one of the key things about the app will be using location information, and while there are 900 species in the country, there's 100 or so within a 2-hour radius of Sydney. And for instance, if we were standing in Hornsby at one of the local reserves, there might be only 10 species around. So the app's able to cut out and discard the species that aren't even local, and just by knowing where you are. And then there'll be a few key points you can tell the app. Is the bark kind of white, or is it kind of stringy and brown and rough? And by that, the app can really break that down from 900 to 1 or 2, just in a few. So that'll be the power of the app, cutting out all those unwanted species. So Anna, that'll be good for you when you're doing your tree identification, huh? Another feature with eucalypts is their juveniles, or their baby leaves. So they actually look quite different to the trees we see around us, and that's a photo shot of some Angophora. These have been germinated in Hornsby Nursery, which is an amazing place if you haven't checked it out, and they have plant giveaways every year, so I highly recommend that you head along there for those. They've grown probably about 5,000 seedlings for me to date, and of those 5,000, the volunteers and Ross, who coordinates the nursery, hand-picked like the top 1,000 seedlings, and they've been potting them up and growing them along. And here they are. This is them on the left. They're in a greenhouse, and they're getting quite pampered because they have nice warm air. They've got humidity sensors which come on if the light's too high, and they have heated bed. It's kind of like a little electric blanket for them underneath running, so they grow super fast. On the right is a picture of the volunteers' work where they potted them up. So there's about a dozen or so species there in those pots. And then once they've sort of fledged their greenhouse nest, they end up outside where they get exposed to all the elements, the sun and the wind. And they're actually quite fast to grow. To get them to about 50 centimeters tall, which is big enough for planting, it takes about 9 months. And from the initial process of laying down the seed. So they sow the seeds, 1 week later they're popping up, and then sort of 9 months later they're ready to go on the ground. Okay, and the photo shoot. So here we have this Angophora looking all pretty. So I've taken this one home, and again got the scale out and everything, the ruler on the side. So I'll be taking photos of the juvenile leaves to put in the book, because you can often tell them apart just by how their first set of leaves look. Okay, this is just a few places I go on my travels. This is Mount Clyde in the southern tablelands. This is it after all the bushfires, when the fog's just come through. So that's why the trees have lost a lot of their leaves here. They've been really cooked. Yeah, I've seen, I absolutely love going out and doing the field work. The computer work, when I get back and processing everything's a little bit meh. I can sort of take it or leave it, but being outside and cooking on the fire and having, gliders shoot across my camp, and gang cockatoos, which I think they're back in Hornsby now, coming through, eating the gum nuts, snakes and wombats. There's everything out there. This is an area near Nara, which has a couple of rare eucalypts, which are only found in that area. This is a few shots after the fire. You can see on the left, there's one of the eucalypts is starting to shoot back from the base. So they have that capacity to recover from fire. Most of them can, some of them haven't. The fires have been too hot. This is up in Putty Road. Again, the fires are just, well, think what it would be like if you were a koala, you know, nothing to eat there really. And no nectar produced for the birds. So yeah, very hot. No leaves left again. On a more happy note, this one is doing very well. This is the tallest tree in New South Wales. It's over 70 metres tall. It's called the Grandis. It's probably my present favourite tree at the moment. And again, this is very old tree, old growth tree. And this is what the forests used to look like. So yeah, it's beautiful. I think just to wrap it up, I think my advice on how I got through science and what I would give to people looking for a career in science is, like Nathan was saying, to do something you love. And I've found setting the goals and being quite dedicated to follow those dreams has paid so much dividends. I do this project 5 days a week, sort of unpaid really, and I just love it and I don't want to stop doing it. And when I think back to my humble beginnings, when I was trying to choose what line of work or career to get into, I had no idea. So for people who are in the audience perhaps have no idea, I remember being at high school, trying economics, hated it, that was good. Accounting, also hated that. Sorry to the accountants that may be in the audience, but you know, just wasn't my thing. But that I learned a lot from doing those lessons of what not to do. And yeah, I think also when I look back at my grades, I worked hard, but they weren't very good. But it was, again, that dedication to studying and doing things I wanted that eventually got me to where I am. So, yes, I think I'll leave it there. Thank you.
Thanks, Paul. Well, if I can see the flowers of the eucalyptus that close, then I think I will be able to identify them.
Oh, nice.
See the meters up. That's my problem. So our fourth speaker, it's Associate Professor Olga Shimoni. She's a researcher on nanotechnology-based tools for biomedical applications. She's as well at UTS in the Faculty of Science. Thanks, Olga.
Hi, everyone. And it's a pleasure to participate in this panel. And thanks for the invitation, Anna. I hope you enjoy. And I want to tell a little bit what I'm doing and what type of scientist I am. So to start with, so I'm experimental scientist, but to start with how I got into nanotechnology. Well, since I was in my teens, I was really into sci-fi movies where those tiny bots traveling through human body, shooting, killing cells or repairing cells. So I was really fascinated about that. And really, that's what brought me into nanotechnology. Well, when I started at uni, there wasn't such a field to study nanotechnology, and nanotechnology is actually doesn't exist in silo. It sits on interface of chemistry, physics, biology, material sciences, engineering, and many fields. So I started with chemical engineering, with more focused on micro nanotechnology. For my master's, I moved into materials engineering, that I studied some polymers, chemistry, polymers, and nanoparticles. And eventually, for my PhD, that's where I moved to Australia. And I did my PhD making some nano-engineered capsules for targeted drug delivery. So that's brought me these days what I'm interested in and what I'm doing. So what is nanotechnology and where we're on a scale of nanotechnology, just to bring a perspective what I'm talking about. So if we start in this space, so there's my laser pointer. So this is a football ball. So that's our scale that we can see and interact. Pretty much smallest part of our body, we can see that's a diameter of human hair, and nanotechnology is 10,000 times even smaller. So when we're talking about viruses, and now coronavirus, it's very hot topic, and we all know about this. So coronavirus is around 100 to 150 nanometers. It's in this scale, and that's basically the scale. So between 10 nanometers to a few hundred nanometers scale, that's what I do my research on. So viruses, very close to my heart in the scale what I'm talking. So why nanotechnology, and why it's important to go so much into such a scale to study it. So if we take a cube, we can see what's the volume and how much surface area of 20 and 20 centimeter. We divided this cube by 2 on each side, so into 8 smaller sides. So we divide by 2 in order of 3. And if we keep dividing every time, every part into 2, and eventually to get into nanometer scale, when I'm working, we divide it by 10 into order of 21. So if you look at the surface of original cube that we started, it's a lot of surface. But if you look at the surface of all these nanocubes made of this big cube, this surface is 10 into 24. It's 10 million times larger than original cube, surface of the cube, not volume, but surface. So that's the surface that we can use to interact with other materials. Materials that I'm talking is in biology. So my research is working with nanoparticles, and I'm working with different type of nanoparticles. Some of them jewelry nanoparticles, it's nano diamonds or nano gold, but some are more like silica, which is typically nano sand particles or other types of particles. And I apply into health technology. So if we have diseases and disorders, I look at what type of nanoparticles can we apply and create advanced health solutions and technologies. And technologies that I'm talking is in terms of diagnostics, how we can identify and detect diseases. It's about the targeted drug delivery, so therapy treatments for cancer, which can be more precise and much more powerful than current anti-cancer drugs. Or field in imaging, where can we look at the cellular structures using nanoparticles and many more. So why into health technologies? So because of the surface of particles, so much surface, we can interact with different types of cells and inside the cells, and we can identify those biomarkers for disease, and we can detect them when it's only a small amount available, so we can identify diseases early. So I worked in the field of cancer, and you can see if 5 years survival from cancer is largely depends when you detect it. If it's in the early stages, you can very well survive and live post-cancer up to 100% in stage 1. But right now, typically cancer detected in later stages, it's when the tumor already developed and sometimes metastasis started, so survival chances are really late. Also, a big chunk of my research is into Alzheimer's disease and brain sciences. We all know about dementia or Alzheimer's disease is 50% of cases of dementia. And dementia is currently identified when people start having memory loss and really showing cognitive decline symptoms. That's where Alzheimer's disease already irreversible. So I put a lot of my efforts and attention to how we can identify Alzheimer's disease before all these visible symptoms can be seen. So actually Alzheimer's disease started 20 years before actual memory loss symptoms detected visible. So 20 years before development of dementia, how we can identify that. And nanotechnology helps us in getting those clues about what's happening before. I told that in my PhD I worked on nanoparticles. So this is like small nanocapsules for targeted drug delivery, and why it's important to work on, because right now when we inject the anti-cancer drugs, it would go to the whole body, causing toxicity to healthy cells. So we, with the help of nanotechnology and in nano-engineering, so we can put specific targeting molecules on surface of this nanomedicine therapeutics to target specifically to the point where the cancer and kill an image or to help in diagnosis of cancer specifically to that particular area. In the detection of Alzheimer's disease, I work with some luminescence nanoparticles, and you can see this is the cell, like healthy cell, and those blue dots, it's actually nanoparticles that you see, single nanoparticles. You cannot see single nanoparticles by himself, but the light that they emit, think about as a beacon or as a lighthouse that emits light to the 100 kilometers to see. So these nanoparticles is the little lighthouses. They're so bright that we can see them with the naked eyes. And with help of these bright nanoparticles, we're trying to identify biomarkers in the eyes that's connected to brain on the back of our eye in the retina. Because of connection to the brain, we want to identify these earlier biomarkers for dementia. That's more. And now in the era of COVID, we had to pivot, me and my collaborators and my co-workers, we had to pivot and put some efforts in identification of COVID-19 detection kits. So currently, there are two main streams in detection of COVID-19. One is based on detection of antibodies in the blood after you get infected, and a week or so after your body starts developing antibody against virus, and you can detect in this sort of like a pregnancy test devices. This is fast, but problem is it's not sensitive. And currently, all the clinics, when you go to test it, get tested for COVID-19, is taking as this nasal pharyngeal swab, so swab from your throat and your nose, which is very, 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 very unpleasant test. And I know I got tested just last week. And what we develop sort of another this test, like a pregnancy test, but instead we're using these bright luminescent nanoparticles and creating much more sensitive. And instead of detecting antibody, we're actually detecting the virus by itself. So it's a 1,000 times more sensitive than current this sort of antibody test. We're detecting virus instead. And we can detect with sort of like a mobile phone camera, or we're actually working with some industrial partner who has monitoring device. So it's sort of, you take a bit of your saliva, you lick, then press, and the monitor shows you, do you have the virus or you don't have. So that's something that's exciting, and hopefully in next 6 months we'll start seeing that's going into the market, into regulation. So that's for me, and welcome any questions from everyone. Oh, I forgot. That's another application of oops, of nanotechnology that's we created using gold nanoparticles, so little golds, which gold nanoparticles are not as gold as you think. They're actually red like this because of the light absorption, interesting light absorption that they have. And we developed tests for easy screening and detection of celiac disease, also known as gluten intolerance. So we all know about gluten-free diet, but gluten-free diet is essential and saves people with celiac disease. It's not a fat diet. It's nothing healthy for normal people, but people with celiac disease, it's a life-saving diet. So current diagnosis for celiac disease is basically a blood test, or gold standard is actually biopsy of the small intestine here, and the general anesthesis. So it's a lengthy and really complicated process. So what we developed, so just by spitting into tube, mixing, and we can see within 20 minutes if you have celiac disease or you don't have, which is a affordable, quick, and completely non-invasive detection test. So we were trying to commercialize this test before COVID, but then COVID happened and we couldn't get into clinical trials with that. But hopefully we'll get soon back. So thank you.
Thanks, Olga. Amazing, all these applications that we can do. I might start with a question for you. So to do what you do you need to study medicine, or to come from more like a technological, I don't know?
No, you don't need to start with medicine. You need to start with science. So these days, and at Australia and Sydney, there are already streams that directly lead to nanotechnology. So it's a part of science. It incorporates a bit of physics, chemistry, biomedical sciences. So it depends what pathway of nanotechnology you would take, and can lead for some amazing discoveries.
Right. So we are going to start then the Q&A session. I will try to pick some of these questions because some of them are really good. I don't know if we are going to be able to get through all of them. But we might start with one that is generic, and then we will move to some, more specific to your topics. There was one about what advice could you give to someone that is in year 12 and it's deciding what to study in university or what to do, when it grows up.
I actually wrote a bit of an answer already in the chat, but I think, as lots of people have touched on, ideally you want to do something that you really enjoy. You spend a lot of time at work, and if you don't enjoy what you do, it's going to be a bit of a grind. And it's much more fun if you enjoy what you do at work. So, but the thing is that when you're in grade 12, knowing what you're going to enjoy doing for the rest of your life is, well, some people do. I had friends in high school, I'm going to be a doctor, and they went and they did medicine and they became a doctor and they're still a doctor and they love it. But I certainly wasn't one of those people. I had no idea that what I was going to do when I chose my undergraduate degree, which was science and arts as a parallel degree. I basically chose something that would allow me as much flexibility as possible to do like different things and work it out. And really, even as I went along, it was a case of, still not sure. Even at the end of my undergrad degree, I wasn't convinced that I wanted to be a physics researcher, though I was kind of narrowing in by that point. So often at high school, I did a lot of tutoring of high school kids. There's a lot of pressure. High school students feel this, the parents feel like this is the most important decision you'll ever make in your life. This is the decision that will define all your future successes or failures will result from this decision. And it's just not true. It's just, there are many opportunities to change, move directions later on in life. And the fact is that at high school, you study such a limited range of choices of what you can learn about. And at university, there's so much more choice of what to learn about. And in life, there is so much more choice about what to learn about. So it's quite you may have no idea. So choose something that, gives you the flexibility to experiment and explore around. Don't be afraid to change your mind and move on to something. If you find something that you really love, and then do it. And even if you struggle at school, I know a lot of people who struggled at school, didn't really get it, really worked hard, but, struggled. And they got to university, suddenly discovered, this is something that I actually want to learn about. All of a sudden, being good is much easier when you enjoy what you're doing.
Yes.
It's so much easier.
Thanks. Does anyone else want to add on, or probably agree with what?
Yeah, I think what Nathan was saying, I went through myself. I had no idea what I was going to do. I even went to the uni open day. We went on a bus. It took us 7 hours to get there. And at the open day, I kind of skipped it and went to my friend's place. So I had no idea what was uni was happening when I got there. So, but it all fell into place. I started studying sort of earth sciencey stuff. And then eventually got really inspired by one of the lecturers in environmental science. And that's what really captured me. Like Nathan was saying, you sort of start broad, particularly in science. You don't have to choose your final major. You can jump around a little bit as you go along. And, if the degree is 3 years and you decide after 6 months or a year that you want to do something quite different, in the grand scheme of your education, your life and your working career, adding 1 more year at uni is nothing. So I just, you'll figure out what inspires you, and don't be afraid to take a little bit longer to try different things. I first enrolled in earth science, a computer, and a psychology course. And then the dean called me in and said, you can't get a degree doing such different things, and made me choose one. But got there in the end.
Very good. So I might move into one more specific question. Bethany, I can see here that someone is asking, what did you find out about the IOD? Has it changed over the last 1,000 years? And can you please explain what is the IOD, or what it stands for?
Yep. So the IOD is the Indian Ocean Dipole, which is the climate system that is based in the Indian Ocean basin, so over to the west of Australia. It's similar to El Nino and La Nina, and you often hear those terms. So we're sort of almost in a La Nina, which is a very wet season for Australia. The IOD works in a similar way, where it has a positive phase, which leads to dry environments across Australia, and it has a negative phase, which leads to wetter conditions across Australia. So over the last couple of years, we have seen a few positive IODs. Last year was a really strong positive one, which led to a lot of drought across Australia. And this was a precursor for the bushfires that we saw. We just hadn't had much rain at all last year or the year before. Over the last 1,000 years, we are seeing that the frequency, so the occurrence and number of events, is becoming more. So we're getting more of these events occurring. They usually occur between July and December, and we're seeing them more often than we have historically in paleoclimate records. And we're also seeing that they're stronger. So strong events have occurred very irregularly in the past, but we are now seeing very strong ones occurring more frequently. So 1994 was a strong one, 1997 was another strong one. They're the two strongest, and then last year was the third strongest on record. And these have a lot of consequences for Australia's climate, and hopefully, well, it'd be nice to not have another strong one, but it's unfortunately looking like we might have another one, so we might get more bad bushfire seasons in the future.
Yeah. Thanks very much. There was a question that it was, they mentioned your name, but I think it's for everyone. There were after tips for young professionals or researchers to stay on science or to stay in a project for long. You know, I don't know if it comes from someone that keeps, you know, not being able to stay in science for long periods, or if it's something about.
Well, yeah, I guess a PhD. So mine, I was actually doing research for about 4.5 years, and then it's been about 12 months of waiting for it to all come back and everything. The nice thing about my PhD was it was still quite diverse. Like I had sort of 4 sub-projects within the PhD. So even though it was a long project, it's been broken up into lots of smaller projects, and every day is a different day. You don't really sit there and do the same thing weeks on end. It's very diverse and there's a lot going on. But then I've only just finished a PhD and probably don't have as much research experience as some people who have probably worked on longer projects over, bigger periods of time. But I found my PhD quite manageable in that it was broken up a lot into smaller projects. And I just really liked collecting the data and finding out something new about the planet we didn't know already.
Is there any other advice from the associate professors to young, or to early career people?
Do you want to say?
I guess to stay focused. Like Nathan already said, if you love that, then you stay focused and you stay on that topic. So you're just fascinated to learn more and more about that, and that's what keeps you focused.
Mm-hmm.
I think there's another really important thing, which is, so in some sense, a PhD is kind of a luxury time in your research career where you have like this extended period to study some sort of task. And as Bethany said, many PhDs are broken up into kind of a collection of smaller projects that kind of lead on from one to the other. But nevertheless, it's kind of a period which is has a bit more freedom, much more so than if you go on and become a postdoc. Often you've got 18 months or 2 years and you have to be in and out, and you know, it's much more time pressure. But at the same time, this can look like a lot of people go through the kind of the PhD blues at some point. I think one thing that's really important for trying to stop that, I mean, Olga has said, you know, choose something you really love. That is important. And the other thing is try to keep some sort of balance in your life. Don't just do your PhD to the exclusion of everything else in your life. Because if you do that, then at some point you wake up after 2 years or 2.5 years and you're just like, what, I don't know where I've been for the last 2 years. I'm still, things aren't working. It's just everything is difficult and everything becomes hard then. So if you keep some balance, that's really important. And the last thing I would say is really important is don't expect science to be something where you're like, here's where you start, that's where I'm going, I'm just going to go in a straight line, and that's how I get there. Science doesn't work like that. Science follows a meandering path. You kind of have to follow your curiosity, follow where the research takes you. And sometimes it takes you in very unexpected directions, and you kind of want to try to guide it. So you always keep your eye kind of on a target somewhere. But if you think that science is going to be like a straight line, then you're going to get very, very frustrated and depressed when it doesn't work that way. And it's not how it works. And in fact, that's not how the best science happens. The best science often happens as you kind of just deviate off to one side and explore this thing. I mean, Olga already gave an example in her presentation where they were doing something completely different, but all of a sudden were able to pivot their research to start, you know, doing COVID testing.
Yeah.
And that's that. So don't expect science to travel in straight lines. It's not how it works.
I agree. So Nathan, there were a couple or quite a number of questions about that. I don't know if you want to answer them or we can move in relation to your topic. There were things about polar graphs and yeah. Is there any of those ones that you answered already that you would like to go through for everyone? Or is there any of them that you thought that it was very interesting?
Sorry, I muted myself again. Yeah. I think one of the things is really interesting is why is Sydney, a couple of people asked why is Sydney a global leader in quantum research? And in some sense, it's really a case of on the shoulders of giants. Like Australia had some pioneers in the field from the early and mid 90s who were working in this area, and they started doing like world leading research from Australia and New Zealand actually, and they started very early, and some of these people were really special scientists in the sense that they weren't only good scientists. They weren't, I mean, they were like someone like Jared Milburn, for example, someone has been a mentor to me. He wasn't just a great scientist, he was also a community builder. And you get some people who really make an effort to build a community and to grow what's going on. And so that created an environment where Australia started investing in this area very early. Like 20 years ago, when I started my honours degree, they created the first big centre in quantum computing technology. Back at the time when people were saying, yeah, but okay, realistically, quantum computers are probably never going to work. It just probably doesn't, it's going to break for some reason. We don't know why, but it's going to break. And they started investing in this first big centre. This was much before other places were doing that. And that investment, that's been continued and it's grown, and people have come in and they've looked for great people, and great people bring in great people, and it builds into great science. And we have kind of a head start, and we need it because, you know, Australia doesn't have as much money as other places. So now other places are like throwing lots of money at things to try to catch up. But in some ways we're still ahead, and we still have really unique, absolutely groundbreaking leading research happening here that really is ahead of everything everywhere else in the world.
That's good to hear. And so I have a general question that anyone can answer. It's one of the last ones. What have been the downsides of working in science? And is it difficult to find work in science?
There's downsides, I think, to every job. Sometimes you do get stuck in the lab working really long hours because you need to get an experiment done and it's time sensitive, so you really just need to get it done. Sometimes your experiments just don't work and you have to start again, or you have to restart your mass spectrometer run because it crashes, or you can't get the samples that you want to get. We have to get permission from the Indonesian government to go over to Indonesia to then take the coral samples. That hasn't always worked for us. Sometimes they've said, no, you can't come in and get the samples. Yeah, there's a whole lot of things that can go, particularly if you're field-based researchers.
Anyone else?
Yeah, that doing a career, and you have to love it, and you have to love it partly because doing a career in science can be hard, especially once you go beyond. You know, kind of getting into a PhD program is that's one thing, but when you go beyond and you try to do your further research, there's a lot of traveling. That sounds great, yay! But moving country is actually a really hard thing to do. Like it's not that traveling to another country is fantastic and a lot of fun. Moving your life to a new country and starting with the bank accounts and the, you know, working out a new admin and a new language and a new system, it's really hard and exhausting. So meeting new friends and all this sort of stuff. And so when that happens and it goes on quite slowly, quickly, only have these short periods in simple places. Sometimes you don't have contracts that go for, you might stay somewhere for 2 years, but you only had, the longest contract you had during that time was 9 months or something like that. And this can be very unsettling. And so I think this is a real tragedy of science, because science is so important, and we need as many different voices in science as we can, different types of people doing it, bringing different inspirations to it, people from different backgrounds. And when the path of a research scientist ends up being difficult like that, a lot of people give up on it. And you're more likely to, the people who are most likely to give up are the people who don't quite have the same privileges as other people, who maybe don't have a lot of money in their background to fall back on, or a lot of women end up giving up earlier because, it's just, they want to prioritize. You have to be a bit egotistical to get through that. And sometimes, and not everyone wants to do that. And so we lose a lot of really talented people from science because we're not creating we're not creating an environment where scientists can flourish with that. And the thing is, a lot of scientists, there's a lot of senior scientists who go, oh, well, I did that. So, if I manage to get through, then, people who are good enough should be able to get, but there's a lot of luck. People tell a lot of success stories about the way you have to be a scientist is to do this. But what they really mean is the way you have to be a scientist is to do exactly what I did because that worked for me. But there are a lot of paths to get there. And we have to support different types of people. And if we don't, then it makes it harder and harder. And we really restrict the voices in science. And it's something we need to work on as a society on how to get better. And some countries do it better than others.
Yeah, I agree. So, Paul, I have here a question about what you do. I don't know if you want, if you want to answer it, but it says, do you think eucalyptus trees reduce biodiversity since they can survive harsh conditions?
So they've reduced biodiversity?
Yeah, because they can survive harsh conditions.
Yeah, look, I think the eucalypts and the dominance in the Australian bush goes right back to sort of climatic shifts. There used to be a lot of rainforest around even Sydney area, like we're talking sort of millions of years ago. So really the drying of the continent and the increase in fire has really promoted eucalyptus, and even over the last sort of 2 million years, the abundance of eucalyptus really within that group is really diversified. And also within the forests around here, partly to do with the geology and the actually nutrient poor soils, the diversity around here is actually really high. It's almost the same as what New Zealand has in terms of its whole plants. So this combination of eucalypt forests and geology that's kind of sandy and kind of poor and fertile, plus the fire, has really diversified things. Actually, there's so many plants and then so many different insects and animals interacting with them. So Sydney's a very diverse place. Yep.
Yeah. Olga, there was a question for you about how do nanoparticles navigate towards a targeted area, for example, like a tumor?
Yeah, I tried to answer, but yeah. So you basically can attach targeting molecules to nanoparticles, something like antibodies or some peptides or amino acids, and they interact specifically with their counterparts that's only found or really overexpressed, or a large amount of their counterparts found on the surface of tumor or tumor cells, cancer cells. So when nanoparticles traveling through the blood system, they found these counterparts on the tumor cells and are staying there. So basically accumulating. They will travel through the blood, but eventually they found the tumor sites and start accumulating that. And the tumor had no choice. Just by uptaking these particles, and the particles can release drugs or have some other therapeutic or diagnostic purposes, and that's how much high amount of particles targeted into one specific area.
Very good. We are getting close to when we need to finish, but I would like to ask everyone, all the panelists, I have a question here that is, just lost it, 1 minute. If you could start all over again, would you stay in science? And if you stay in science, would you change disciplines?
I wouldn't choose different type of science. If I'll start again, still nanoparticles fascinate me until today. But like Nathan mentioned, there is a lot of current struggles for being scientists. So if it would happen, I would probably go to industry to use nanoparticles there.
What about Bethany?
I think I'd stay in science, but it's events like this where I hear about all the cool science other people are doing, and then it makes me want to do all the other sciences. I don't know what I would do again, but I'd definitely stay in science. I probably would have done a bit more diverse subjects in undergrad to learn a bit more about all the sciences. But yeah, there's just so many fascinating areas of research and science out there. It'd be hard to choose something else.
What about Paul?
Yeah, I think as I went through my undergrad, I had to make the choices as I was going along, which, I went to geology, and I knew I didn't want to get into the oil industry, and, various choices like that. And I ended up, the last part of my studies was in New Zealand and partly in Sydney. It was for conservation biology. And that was just really, really what I loved. I loved this idea of working with the forests, the plants, and the animals, which I can see, which are declining, So no, I'd stay in that. How I'd do it would be probably different, because I did my undergrad and a master's, and then I went and worked in the bush, and then they're like, okay, now you got to get a TAFE certificate. And then I got that one. I sort of forced into it. I was like, I've already studied 4 years, but because it wasn't specific to the industry, I had to get that. And then I actually loved it, and I did another year, and then another one after that. So I probably would have reversed it in the other way around had I known, and worked in the bush while I was studying. And then even that can even take time off the uni degree. So instead of sort of doing 7 years, I might have really condensed that down. But I still love it, and I still volunteer, and still are there doing my career that I get money for part-time and do my other project, full-time. So I love it so much.
What about Nathan?
So I love doing science. I think I would have great difficulties at this point going and doing a job where I have to do, I have someone telling me what to do every day. But there are challenges to it, and I guess maybe there are places where I'd look back and think, oh, okay, if I chose a slightly different way, or thought about certain things in advance, then maybe kind of that might have been make things slightly a bit easier, at least in terms of the life. But I think the only thing that I would really think seriously about changing, because I love the field that I'm in and I'm really excited by it. The only thing I would seriously think about changing now is if I was as conscious of the issues associated with climate change and as aware of how fast things were going to happen in that space 20 years ago when, or longer ago when I was in undergrad, then maybe I would have moved in a slightly different direction to try and contribute to that problem. There comes a point, I know people in my field who switched. There's a guy who won a Nobel Prize in the field in 1996. Or no, I think his Nobel Prize was after that, but he did a very famous thing in 1996. And he switched and started like a climate change research center in the US, and he's been incredibly successful. But there's kind of a large chunk of your career where that's not possible. You can either switch quite early, or you can get really, really successful and amazingly important, and then you can kind of switch and do whatever you like later. But there's a kind of a whole gap in the middle where it becomes very difficult to dramatically change direction. So that maybe would be the only thing I'd think about.
Okay, very good. Well, I think that we need to close the event here. Before we all go, I first of all would like to thank for the time of the panelists. I didn't meet any of you before, and it's been great to meet you through this event and to know about you. They are all local residents from Hornsby, so you might see them around Westfield. So say hello and ask all your questions. But yeah, thanks for your time. Very generous as usual. Scientists are very generous with their time in general. I want to thank as well the Hornsby Library staff for organizing this event. Apologies for that little hiccup technological issue that we had at the beginning, but lucky it was really early on in the event, so we were able to start again. And just keep an eye on this kind of event for next year's, because we are definitely happy to host another event during National Science Week. We did one last year, we've done one this year, and we will put our thinking hats on to think about doing something next year. So please keep an eye during this week, because something will come up through the Hornsby Library events, and make sure that you look at any other events that this library organizes, because it's always amazing the amount of things that they do here, different topics, different subjects, and all very interesting. So I'm leaving it here. Keep enjoying National Science Week. There is plenty of events out there. And thanks everyone for joining from the audience and from the panelists.